Hi, my name is Daugherity. I'm a clinical fellow at Cornell University. Hepatobiliary cancers are common, costly cancers that carry a poor prognosis. Hepatocellular carcinoma is the 5th most common cancer in the world. The incidence of hepatocellular carcinoma has doubled in the past 2 decades, and it's one of the only cancers increasing in incidence in the United States, despite a decrease in other risk factors, including toxin exposure and hepatitis virus infection. This is thought to be due in part to the emergence of a spectrum of fatty liver diseases, the most benign being non-alcoholic fatty liver disease, and at the upper side of the spectrum, the more malignant non-alcoholic steatohepatitis, or NASH. The prevalence of fatty liver diseases are increasing in the United States, with 13% of new hepatocellular carcinoma cases occurring in patients with fatty liver disease. So, it's known that hepatic fat accumulation contributes to hepatic tumorigenesis. However, the interaction between fatty liver disease and cancer induction is currently not well understood. We hope to establish an animal model capable of evaluating the interaction between fat accumulation in the liver and hepatic cancer. We hypothesize that DNA is damaged through the generation of reactive oxygen species during fat breakdown, contributing to hepatocellular carcinoma. More specifically, excess free fatty acids from a high-fat diet are metabolized, leading to excess free radical release. This damages mitochondria, hepatocytes, and they react with lipids to form highly toxic peroxylipids. This, in combination with inflammation, causes oxidative DNA damage. When DNA repair pathways are intact, this DNA damage, in the most part, is repaired. However, when DNA repair pathways are not intact, the DNA damage may not be repaired, which could possibly lead to apoptosis, fibrosis, or carcinogenesis. It could also lead to protection against these entities, and this is what my project focuses on. In eukaryotic cells, both cellular metabolic activities and environmental factors damage DNA. Examples of exogenous sources of DNA damage include UV and gamma radiation and toxins and chemicals. But endogenous processes can also damage DNA, including replication stress, and more importantly to this project, metabolic byproducts, including reactive oxygen species. Eukaryotic cells protect genomic integrity in part through checkpoint mechanisms, including a major pathway centered around the protein kinase ATM, or ataxia-telangiectasia mutated. ATM is recruited and activated by DNA double-stranded breaks. It then phosphorylates downstream substrates such as CHEK2 and P53 and ultimately leads to cell cycle checkpoint arrest, apoptosis, or DNA repair. ATM is a very important factor in protecting the genome against DNA double-stranded breaks. In fact, when mutations occur in the ATM gene in humans, a genomic instability syndrome called ataxia-telangiectasia occurs. These patients have an inability to deal with programmed double-stranded breaks that occur during meiosis, immune system maturation, and telomere maintenance. There is a mouse that is null for the ATM gene due to a targeted mutation. These mice are exquisitely susceptible to oxidative DNA damage and exhibit similar phenotypes to the human syndrome, such as cancer predisposition. That is why the ATM null mouse was chosen as a model in this study. ATM null mice and control mice were fed either a standard diet or a high-fat diet for 8 weeks, and tissue was collected. After confirming that the high-fat diet does cause steatohepatitis, hepatic oxidative stress, and DNA damage via histology and reactive oxygen species and DNA damage detection, we found that 8 weeks of feeding the high-fat diet increases the rate of ATM-dependent steatoapoptosis and causes other phenotypes similar to NASH in humans, including hepatic fibrosis. This was done primarily through gene expression analysis and immunohistochemistry. These data demonstrate a prominent role of the ATM pathway in fatty liver disease-related steatoapoptosis and suggest that a high-fat diet triggers DNA damage that induces ATM-dependent apoptosis. This establishes the ATM null mouse as a powerful model to study the progression of fatty liver disease to hepatocellular carcinoma. Thank you, and I invite you to visit my poster at the conference.